number 4 here is begin looking into, DNA fingerprinting. Okay, this is a method of developing a person's DNA profile. It was pioneered by Dr. Alec Jeffreys. The whole idea behind it was that as each human's DNA is unique, that means that we should be able to identify humans by their DNA. It was first used by law enforcement in the mid-'80s in England. The DNA evidence actually exonerated one man and convicted another. There is a book about it. There was also another instance where it was used to prove that actually maternity of a child, which is sort of an unusual thing, but it had to do with a deportation case. It was first allowed to be admissible into a court of law in the United States in the mid-1990s, when we began to figure out that it was actually a pretty consistently accurate methodology. So 99.9% of your DNA is the same as everybody else's. Okay, we have 0.1% that differs. This is a combination of gene differences, so the difference between the alleles of blue eyes versus brown eyes, or the ability to curl your tongue or not, or whether or not you have a widow's peak, right? Those sorts of things. And then there are differences in polymorphic regions between the genes of DNA. Polymorphic just means many forms. Okay, which you'll see what we mean here in a second. Certain points between the genes on DNA have repeating base sequences. If you see here in red, it's the CG repeat, and this is referred to as a short tandem repeat, or an STR. STRs happen at the same place in everybody's DNA, but they are different lengths. For example, here we have person 1 that has 7 repeats, but person 2 has 5 repeats. Well, if you compare those, and you compare all the STRs in an individual, it's easy to identify the differences between them. So, we use our good old friends restriction enzymes that we talked about before, and we chop up the DNA into millions of fragments. Okay, some of the fragments contain STRs, some don't. The ones that do are then different lengths for different people. If you look here, you can see where these polymorphisms are. Okay, so this is RFLP's restriction fragment length polymorphisms. In allele 1, you can see the restriction enzyme is actually going to cut in 2 places, whereas in allele 2, it only cuts in 1 place. And you're like, that's great, but I promise that will make more sense in a minute. Now we use gel electrophoresis. So gel electrophoresis, I use agarose and I dissolve it in water. And then, so you heat it up, you dissolve the agarose in the water, and then you let it cool. And just like Jell-O, it makes this happy little gel. Okay. Now, this can be used to sort proteins or nucleic acids on the basis of their electric charge and also their size. Okay. So, DNA has a negative charge to it. So if I put in an electric field, the DNA is going to naturally migrate towards the positive charge. The agarose gel has little holes in it. When I describe it to younger kids, I'd say it's like steel wool. I mean, it's much softer than that, but there are little holes in that. And it sorts DNA according to fragment size. Just like driving along roads, a motorcycle is small. It zooms in and out real easily. It can get someplace faster, as opposed to a semi that if it's on really windy roads has to go much slower. Smaller fragments migrate much more quickly than larger fragments. So that means I would have started my DNA at the negative end here, which is on the left of the picture that's on the right-hand side of my screen here. And then you can see in allele 2, because it's a much longer fragment, Z did not migrate nearly as far. And then in allele 1, I have actually 3 bands because it cut in 2 places instead of only cutting in 1 place. To make a DNA fingerprint, I finally attach radioactive probes to our short tandem repeats, and that way only the ones with the STRs will show up on the gel. You can do something that glows under UV light. You can do something, if it's radioactive, you can expose it to a certain type of film, and it will actually be able to detect it. That's autoradiography. Okay, I did this, one of the labs I collaborated with, even on basic DNA sequencing, we always used a radioactive probe. We use phosphorus 33. It's nice. It's not ridiculously expensive, and it only has a half-life of a month. So if you spill some on your shoes, you only have to leave them in the lab for a couple of months. STRs are different lengths, so in different people, it creates a DNA photo or a DNA fingerprint. You can look over here and you can see where there are differences in each of these. Forensics, we use this. We take DNA from a crime scene, whether it be blood, hair, semen, any other bodily fluid, and then we can compare it to the list of DNA subjects. So if we look here, this is in a sexual assault case. Ladders. Okay, you'll notice that there are ladders sort of spread throughout this. Ladders are known fragment lengths, and they just think of it like a ruler. It lets you know if your DNA is where it's supposed to be. Okay, you have a control, a positive control. This means that you always know what is happening. You always know that everything is working. You, by the way, they don't show it on here, should always run a negative control also, and that means that nothing has been contaminated. In our known section, we have the victim in the first column, and then we have suspect 1 and suspect 2. And then we take from the evidence, the female cells, the sperm DNA. Now note that they've also taken and included the boyfriend in this because they want to make sure that they eliminate that as a possibility. Okay, if you've seen the episode of The Big Bang Theory where their apartment gets broken into and Sheldon gives them a sample of his DNA so that they can make sure that they aren't including him, but he doesn't include Leonard's because, maybe it's an inside job. So when I look here, which suspect committed the rape? Well, if I look at my evidence, I can see these 2 bands about the middle of the region, and that lines up there with suspect number 1. Now, this also works for who your baby daddy. Okay, this is in fact how we do paternity testing. So if I look here again, you'll see the ladders. I have the mother, I have the child, I have the alleged father. Okay, the child should have 1 band from mom and 1 band from dad. Okay, so if we look at the child, you can see that the lowest band actually lines up with the mother, but then the upper band of the child actually lines up with the lower band on the father. And then when you do this child-alleged father mix, you only get 3 bands of DNA. So yes, this is actually a positive test for paternity. I'd like to point out that in a real paternity test, you use far more than 1 of these. Okay. So this, in this case, we have the mother, we have the child, we have the alleged father. But if you'll notice, like the child and the mother's upper band match, okay, but that is slightly longer than the lower band of the alleged father. And if you look at the child-alleged father mix, it actually is 4 bands, which means that this is not the child's biological father. If we look here at this cute little diagram, I want to know which child is not related to mom. It's a little bit hard to see. If I had designed this, I would have set it up so that mom and dad were in the middle. Okay, but like if you look with mom and daughter 1, they have a band that lines up. Okay, with mom and daughter 2, you have 1, 2, 3 bands that line up. Sorry, I'm pointing at it with my little marker here, but you guys can't see that. With son 1, okay, I've got 1, 2 bands that line up with mom, but with son 2, I've got nothing. Nothing lines up here. So son 2 is not related to mom. What children are not related to the father? Again, you can sort of go through and look, and we have daughter 2 and son 2. So we imply from this that son 2 was adopted and that daughter 2 would have been from a different relationship with mom. I'm hoping that that means that they're not there in age, because that could get a little complicated. Blood stain from a murder scene. We have 7 possible suspects here. This is even more what like a paternity test is going to look like too. Lots and lots of bands. And you'll notice that suspect number 3, if you really compare them, is what looks the same. And this goes back to our previous who committed the sexual assault, and that would have been suspect number 1. Okay. I have a fingerprint from mom, I have a fingerprint from the child, and then I have a fingerprint from 2 potential fathers. Which one is the same? Well, if I look here, honestly, I look up, and the second band in the child actually lines up very nicely with the first band in father 2. So the second alleged father is the actual biological father of this child. Interpreting DNA fingerprints. This is just another one. This one, honestly, the labeling is a little bit hard to see, but child 2 is the one that's actually from a different father in this particular case. All this works because of polymerase chain reaction, or PCR. PCR is what my husband affectionately referred to as the DNA photocopying machine. Fun little story. This is actually was created by a gentleman in California. He was quite the surfer, and somebody who had his, I don't remember what he exactly had his PhD in. I don't remember if it was in biochemistry. I don't know that it was actually in genetics. He wrote a book on it because purportedly he was in an altered state when he actually figured out how to do this. It's a 3-step process of heating, cooling, and replication. It's really nice. The little picture here is a thermocycler. It does it for you. This, even before me, I'd like to point out, I never had to do this. You actually literally had to stand there for 2 hours, like moving your samples from one temperature water bath to another water bath every 30 seconds. And that would have been a royal pain in the rear end. How this works. I can very quickly increase small amounts of DNA to have enough to analyze it. What I do is I first heat it up to about 95 degrees Celsius, and that causes the DNA to denature. Now, you'll note by looking at the picture, it does not cause the sugar phosphate background to fall apart, but it does cause the hydrogen bonding in between the nucleotides to fall apart. I then cool it down to somewhere around, 65 degrees Celsius, if memory serves me right. And this allows my primers to actually bind with the DNA that I want. The primers you just order from a lab. Basically, you take a little bit further out from where your gene is, or the region of interest, rather. It doesn't even necessarily have to be a gene. And you say, okay, here's about 12 bases. I want the matching pair to this. If it's ATCG, you're going to want ACTG. You're going to want TGCA. Okay, you're going to want the opposite of it, because remember, they run anti-parallel to one another. And so then that will come in and bind. And then at about, you go to about 72 degrees Celsius, and then that allows the polymerase to come in and then add the DNA nucleotides. We do not use human polymerase. It is well denatured at that temperature. We use something called Taq polymerase, which comes from a bacteria it's a Thermicus aquaticus, which is a little bacteria that lives in the volcanic vents at the bottom of the ocean. It's expensive as sin. Anyway, you go through and then you will repeat the process. You heat it back up to 95 degrees Celsius, and that causes the DNA to fall apart again. And you keep doing this, and you'll notice in cycle 3, I now have 2 pieces of DNA there in the white boxes that are exactly the pieces that I want. You get somewhere around 1 million pieces of DNA. This is an exponential process. Okay, and we will stop there, and we will start with genomics with the next one.